So, hello, biology. Today we're going to be talking about doing the notes. So, if you're absent, the notes for Unit 5, Section A, which is the molecular genetics, Part 1, objectives A and B, covering protein synthesis. So, as you kind of follow along, then you can take your notes and stuff like that as you listen. So, the first part is describe how DNA stores genetic information. So if you look at the DNA, remember the DNA has three parts. So each nucleotide has a sugar, a phosphate, and a nitrogen base. Okay. The nitrogen bases, if you were to look at this and you say, okay, so where would the code of DNA be? The code is actually found in the sequence of these nitrogen bases, as you'll learn in protein synthesis, is really the order that these go in. So like TGCA, the order these nitrogen bases in is what really codes for proteins, which is what builds you. Okay. Um, so you need to understand that really when what this comes down to is your DNA contains the codes that make enzymes. And remember, enzymes are proteins, and proteins are made of amino acids. And that everything in your body is done by enzymes, that no matter what, you have to have enzymes to break things down, to build new molecules. You have proteins that are structural, that build you and make you who they are. So therefore, really, DNA controls everything in the body. And that becomes really important to the fact that they actually called that the central dogma, that DNA is the secret to life, that if you understand that code, the order of those nitrogen bases is so central to life and is the code for everything, that is the central idea or basis for everything that is living. So they call it the central dogma. That DNA codes for life or codes for proteins, which is what builds and breaks down everything in your body, runs all your processes, builds all of you. So, um, the DNA code is in the sequences. It's in the order of the bases. So if you change one of the base sequences, as you'll learn, then you essentially change the code, which is going to cause a domino effect of changing the proteins, which means then your traits or how your body works is going to be different. Okay. So that's really, um, the first part. Now, these four bases are going to code for 20 amino acids. So there are 20 amino acids that build all the proteins in your body. Most of those your body can make. Some of them your body can't. That's why they're called essential amino acids, which means you have to get them through your diet. Remember, we learned that in the macromolecule unit. So, three bases in a row code for a specific amino acid, and we call those, all right, codons. Okay, so a codon is three of these letters together. Okay, so if you start looking at these would be the different amino acids. And for the purpose of this class, you don't necessarily need to know all the amino acids and know what their letters are, but you do need to know that there are 20 of them and these are it. This would be like the code in the DNA. So if you looked in the DNA, the way you use this is you would say if the first letter is A and the second letter is T, and then the third letter would be like TCGA. So if it's ATT, this would be the amino acid that gets stuck in place to build that protein. And this will make more sense when we get into protein synthesis. This is based off of the mRNA. This is the one you'll use more often. You work it the same way. You start on the inside and you work out. So like if it's AG, then it would put in a lysine. If it was UCC, it puts in a cyranine. So that's essentially it. So when you look at DNA, it codes for proteins. Now, if we understand this now, and as you learn about protein synthesis, one of the things we kind of talk about a little bit is this idea of ethics. And where is the line going to be? And there's all sorts of stuff that we can do now. We can um, put different genes from one thing that we found ways to start inserting them into other things. This CRISPR video is a really good link. I also have the link up on my blog. You can also open up the PDF notes, so the PowerPoint, and click on it to go to it if you want to watch it. Very interesting if you missed it. It's some of the really kind of talks and covers some of this ethics part of it. So now that you know how DNA stores the genetic code, which is in the nitrogen bases in the order that they go in, let's look at protein synthesis as like an actual process. So a couple things we need to know. So protein synthesis is how the DNA makes proteins, or the enzymes that run your body. So there are several new players or things that are going to come in play here. There's this stuff called RNA. And there's 3 types. Now, RNA is a ribonucleic acid. It's different than DNA, very closely related, but it's got a different sugar. So if you go back and you remember the sugars there, RNA has ribose sugar, whereas DNA has a deoxyribose sugar. All right, so they both are essentially the same. They both have a sugar, a phosphate, and a nitrogen base. And there's 3 types of RNA that all play a role. One of the other major changes is that RNA is actually single-stranded, which means that instead of it being this double helix, the RNA is actually only going to be one part of the DNA. So it's kind of like half a ladder, okay? The last thing is, because the sugar is different, it doesn't use thymine. So in DNA, you have thymine. But in RNA, everywhere you would put a thymine, instead a uracil gets put. And the main reason is, just in a simplified version, it just doesn't work with the thymine. So you just have to replace the thymine with the uracil, and you'll see what I mean. Now, these 3 types of RNA do different things. So the mRNA is the messenger RNA, and you're going to see that it essentially carries the message from the DNA out to the ribosome where the protein is made. The rRNA is ribosomal RNA. That's where the R comes from, ribosomal. Now, it makes up part of the ribosome. So the ribosomes are what actually build the protein using the messenger. tRNA stands for transfer RNA. Its job is to carry the amino acids to the ribosome so that the protein can be made. Okay? Now, this is a very important video. I would highly suggest that you pause this video perhaps, or when you're done with me going through this, I would definitely watch this video, which is linked on my blog. It will really help visually show you kind of the process I'm going to go over. Like I said, I would maybe even pause this video, go watch this one, and then come back. All right. So protein synthesis essentially has 2 stages, okay? What is called transcription and translation. Transcription, I tell kids, remember, is making a transcript or a copy. So the DNA, all right, is in the nucleus, and this purple part is the nucleus. The nucleus has these little nuclear pores in it. Now, the DNA is too big and too valuable to ever leave the nucleus. So instead, what the cell does is it uses the DNA to make a copy of the RNA, which specifically is called the messenger RNA. This can go through these pores and leave the nucleus, keeping the DNA inside and protected. Okay, that's called transcription, is the DNA making the mRNA. Okay, that's transcription. The second part, called translation, is then the translating of the mRNA into amino acids, or therefore building the protein. So the mRNA is going to build the protein in the second step, called translation. So this occurs in a ribosome, and you have all these ribosomes all over the cell. So wherever the protein needs to be made, that's where translation is going to take place. So transcription is DNA making mRNA. The mRNA leaves the cell nucleus, goes out to the ribosome, and then the ribosome is going to read the mRNA and build the protein, which sometimes is called a polypeptide. That's called translation. It's going to translate this information into a protein. Okay, so those are your two parts. The transfer RNAs are what bring these amino acids in and put them together. Okay, so the codons of the mRNA match the what are called anticodons of the transfer RNA. And this is how the cell makes sure that it knows what to put in. There are also codons that are called start and stop. It tells the enzymes that, or the ribosome, where to start, where to stop. Same thing with in the DNA, you have start and stop codons, which tells the enzyme RNA polymerase, which builds the mRNA, where to start building the RNA and where to stop. So this is just a quick look at it. So here's your ribosome in green. So in the cell, or in the nucleus, sorry, you had a DNA molecule. And the DNA built this mRNA code. Now this mRNA is going to feed through the ribosome kind of like a ticker tape. How does the cell or the ribosome know what amino acid? Well, every amino acid is connected to this transfer RNA that has a specific anticodon. And the anticodon will only match like a puzzle piece with the mRNA. So if the mRNA says AUG, then the ribosome kind of is going to look at this and say, okay, if this is saying AUG, then I need to stick a methionine right there. So what it does is it grabs this transfer RNA that the anticodon can only fit with this AUG. So you always read the mRNA, but the anticodon essentially makes it so like this one could never fit here because we know the base pair rule says A and G does not go together. So the transfer RNA anticodon ensures that the right amino acid always goes with the right mRNA codon. So then it sticks in a methionine. Then the ribosome is going to read the next one. When it reads CAG, then just like before, it's going to go in here and go CAG. It's going to put in a glutamine. So it's going to bring in the transfer RNA that carries glutamine is going to come in and attach right here. And then these two are going to bond together. And then this one's going to leave. The ribosome's going to shift down again. So then what would go in here? You would go, well, GAA, it's going to go and look for GAA. It's going to find a glutamic acid. And then the transfer RNA is going to come in, attach, so that the next amino acid, glutamic acid, can bond right here. And then this one will leave and it'll just kind of keep shifting down until it hits the stop codon and tells it to stop. Like I said, I would highly suggest you go back and watch. If you click on the PDF of the PowerPoint, or you find the protein synthesis video on my website underneath Unit 5, I would really suggest you watch that. It really helps. So if you have any other questions when you're done looking at this, make some time to come see me and we can work through the details.